we move on to Subendu, who is going to talk to us about nitrogen fixation on particles. Subendu, are you there? Ah, there you are. Excellent.
Yeah.
Over to you, Subendu, and thanks very much.
Yeah, just try to share the screen. Yeah, perfect. Hi, good afternoon. I'm Subendu Chakraborty. I'm a postdoc at the moment. I'm a postdoc at the Leibniz Center for Tropical Marine Research. It's in Bremen, Germany. And the work that I'll be presenting, I did this work in when I was in University of Copenhagen, and recently I published it in Nature Communications. And the topic is nitrogen fixation in marine sinking particles by heterotrophic bacteria. So as you know that nitrogen is a very important component for the growth of organisms, and in marine environment, of course, nitrogen gas is available in very high amount, but most of the organisms, they can't use that gas. So they have to depend on the inorganic form of nitrogen, like nitrate, nitrite, ammonium kind of inorganic form. And through phytoplankton, they go to this inorganic nitrogen, so they go to the higher trophy level. And when these organisms die, bacteria, they decompose and convert it into the inorganic pool of nitrogen. But there are also bacteria that can convert this usable form of nitrogen into the nitrogen gas through denitrification. And if it continues for a long time, so at some point, the marine environment will be a desert of this usable form of nitrogen. But thanks to some other type of bacteria that can again convert this nitrogen gas into this usable form of nitrogen. So, and this is called nitrogen fixation. So, but when people measure the amount of total amount of denitrification and nitrogen fixation, they found a huge mismatch between these two processes. So we can expect that something is going on in the nitrogen fixation part that we don't know. Some kind of nitrogen fixation that we don't have any idea, and we need it. We need to know it because it's important for the global nitrogen budget. Now you know that the nitrogen fixation is really important because in some part of the ocean it can support up to 50% of the new production, and as a result, it can play a crucial role in carbon fixation and help to export those carbon to out of the zone. Now, when you think about nitrogen fixation, the first thing that comes to our mind is cyanobacteria. They are actually the major nitrogen fixers in the open ocean, and they're mainly abundant in the tropical and subtropical oceans. But recently, of course, apart from cyanobacteria, there are also people found the symbiotic association between diatom and diazotrophs. They can also contribute to the marine nitrogen fixation. But when people look at the NID gene, which is actually responsible for nitrogen fixation, they found the presence of non-cyanobacterial diazotrophs, that is heterotrophic diazotrophs. And interestingly, these heterotrophic diazotrophs, they are present in all over the global ocean, like nutrient-rich cold to dark ecosystems, including coastal upwelling zone, temperate coastal zone, and as well as in the deep ocean. And this is interesting because the enzyme nitrogenase, which is responsible for nitrogen fixation, it gets inactivated very quickly in the presence of oxygen. So these diazotrophs, they either need oxygen-free environment, or they need some kind of environment where oxygen is really low, or they need to do some kind of trick to cover the cell with some kind of lipid layer, or they need high concentration of carbon to burn all those oxygen inside the cell. But for this non-cyanobacterial diazotrophs, there is not much known about this kind of strategy for them. So the question is then how these heterotrophic diazotrophs, they are abundant or they are available, or these genes are available all over the global ocean. So people speculate that it can be because of these sinking marine particles, that is, they can do nitrogen fixation inside these sinking marine particles. So as you know that these particles, they form of variety of organic matters, including dead animal, plankton, phytoplankton, protist, fecal matters, and other inorganic elements. And the size range of these particles, they lie between micrometer to several nanometers. So because these particles, they are rich with the carbon and other nutrients, so they are basically the hotspot of bacterial activity. And because of high bacterial activity, the respiration is really high inside those particles. And because of high respiration, there can be, of course, it's proven that the anoxic interior can create inside those particles. So where this bacteria can perform nitrogen fixation. But till now, I mean, there is no evidence that, although they found the gene of these non-cyanobacterial diazotrophs inside those particles, but they don't know whether there is no evidence that they are really active inside those particles or not. Or if they are active and they are doing nitrogen fixation, then what are the conditions of when they perform nitrogen fixation inside those particles? Or what are the rates of the nitrogen fixation? Or how significant these rates are compared to the other cyanobacterial nitrogen fixers? So to answer this question, we thought, okay, let's form a mathematical model. And this is a modeling work, of course. So form a mathematical model on heterotrophic nitrogen fixtures, so who are already inside the single particle. So for that, we considered a spherical particle consists of polysaccharide and polypeptide, and bacteria are already there, those heterotrophic bacteria. They release ectoenzyme to degrade those polymers into monomers and oligomers that they can take inside the cell and digest and extract carbon and nitrogen, and they can grow. And if the environmental conditions are suitable for them, they can perform nitrogen fixation. This is the basic model that we have. And if I go a little bit deeper into the model, our model has two different parts. One is cell model and another one is particle model. The cell model basically captures the basic cellular processes that determine the growth and nitrogen fixation in an individual cell. And then we go to the particle model, where we scale up this cell model to the population level to address the particle dynamics and look at the contribution to the total nitrogen fixation in the water. Now, if we think about a particular cell, this is the particular cell, heterotrophic cell, then it has three different resource options, resource uptake options. Like it can get carbon through glucose uptake, it can get both carbon and nitrogen through amino acid uptake, and it can has the possibility to get nitrogen through nitrogen fixation. Now, the carbon from glucose uptake and amino acid uptake, from there they can pay the respiratory cost, and the remaining carbon can go for synthesis. The nitrogen from that they get from amino acid uptake, and if they do nitrogen fixation, they come together and they can go for synthesis. And of course, for respiration, they need oxygen. And in the absence of oxygen, they can perform respiration using nitrate, and further they can also use sulfate. And they affect respiration, and also they can affect synthesis. And this synthesis happens according to the Lebesgue minimum law. That means the minimum of these three quantities will determine the growth of the cell. And the specialty of the model is the cell has the ability to regulate its nitrogen fixation rate based on the conditions that they have in order to maximize its growth rate. For example, as I mentioned earlier, that these diazotrophs, they have the problem with the oxygen because the enzyme nitrogenase, which is responsible for nitrogen fixation, it gets quickly deactivated in the presence of oxygen. And that's why they need some kind of oxygen-free environment or oxygen-less environment. So if the oxygen is really high in the surrounding, so they will not perform nitrogen fixation. But at the same time, if they have enough carbon source, then what they will do, they will use that carbon source to burn that oxygen, and they will fix nitrogen and increase their growth. Yeah, this is the basic mechanism. They can regulate their nitrogen fixation depending on the conditions that they have. And if we go to the particle model, then we have a particle consisting of cells, single cells, and single cells of bacteria. And the particle consists of polysaccharide and polypeptide. And the bacteria, they will release those ectoenzyme to convert polysaccharide into glucose, polypeptide into amino acid. and they use those glucose and amino acids to for their growth, and the excess amount of glucose and amino acid will just diffuse out from the particle. And of course, oxygen, nitrate, sulfate, nitrogen gas, they can also come inside the particle through diffusion. And yeah, so if the conditions are suitable for those bacteria, they will do nitrogen fixation. That's the basic model. Now, if we look at the dynamics of the model, so I have considered a particle of radius 0.125 centimeter. Yeah. And I run the simulation for 4 days. And along y-axis, you can see that 0 means this is the center of the particle, and this is the surface of the particle. So if you go in this direction, that means this is the direction of along the radial direction. So I start the simulation with an initial concentration of level polysaccharide and polypeptide and some bacteria. And outside the particle, I used a constant oxygen and nitrate concentrations. So when there is bacteria inside the particle, they will release ectoenzyme and degrade those polysaccharide into glucose, polypeptide into amino acid. As a result, polysaccharide will decrease, polypeptide will decrease, and glucose and amino acid concentration will increase, and bacteria will also increase. At some point, bacteria will reach very high concentration, and respiration will also be high. And because of high respiration, they will get anoxic interior inside the particle. And when there is no oxygen, of course, they will start using nitrate. And eventually, at some point, they will also finish nitrate, and they can also, at that time, they will also use sulfate for the respiration. Now, when they finish all the amino acid, so they don't have any extra source of nitrate. And still, they have some carbon left, and there is no oxygen inside the particle. So what they will do, they will start fixing nitrogen, and get the nitrogen from there and keep going. But of course, at some point, so the bacterial cell count will decrease, and oxygen concentration will increase, and they will not have enough carbon to deal with that oxygen, and they will stop nitrogen fixation. And that's the basic dynamics inside the particle. But what I considered here, that the oxygen and nitrate concentration, they are fixed outside the particle. But in nature, of course, they are not fixed, right? So when the particle sinks, they face a gradient of oxygen and nitrate. So that's why I have considered like a water column with oxygen and varying nitrate concentrations. Then this is taken from somewhere in the North Atlantic Ocean. And of course, I considered a different size class of particles, because in nature also, I mean, the size also of different particles they vary. So what we found, that the first thing that we noticed, the large particles, the bacteria fix nitrogen at a high rate for in the large particles, and the possibility is also high for them. And this is because in the large particles, what happens when the oxygen comes through diffusion inside the particle? As you see here, the bacterial cell concentration is really high, close to the surface of the particle, and because of that, when oxygen comes from outside inside the cell, they finish all the oxygen close to the surface, and because of that, they have anoxic interior. And in large particles, the possibility of having anoxic interior is really high, and that's why, yeah, they can do nitrogen fixation at a high rate in large particles. And also, the time interval when they can perform this nitrogen fixation is also really very, very tiny, very small. And another thing that we noticed here, that the sulfur reduction is more important for nitrogen fixation compared to denitrification. And this is also because of the same reason, that since bacteria, their bacterial concentration is really high, close to the surface of the particle, and because of that, when nitrate diffuse in from the surface of the particle towards the center, then they finish all the nitrate close to the surface. And because of that, in most of the region, nitrate inside the particle, nitrate can't reach. And because of that, the bacteria, they perform nitrogen fixation using sulfate. in most of the parts. And that's why we found that maximum 6% of the volume of the particle, they can perform nitrogen fixation through denitrification, whereas more than 80% of the volume of the particle, they perform nitrogen fixation using sulfur reduction. Now, of course, in nature, the different types of particles, they exist. To mimic that one, so we consider 3 different types of particles, and they vary in their sinking rate. Like here, we have considered marine snow particle, diatom particle, diatom aggregate, and cuculithophoid aggregate. Where cuculithophoid aggregate, they have very high sinking speed compared to diatom, and marine snow, they have very low sinking speed. But you notice here that the region within the green dotted line, this is actually the hypoxic region where the oxygen concentration is around 62 micromole per liter. But we see that in natural marine snow and diatom aggregates, most of the time they fix nitrogen within that hypoxic region. But because of very high sinking speed of cuculithophoid aggregate, they fix nitrogen outside that hypoxic region where oxygen concentration is really high. So we can say that the anoxic microenvironments as well as nitrogen fixation can extend into well oxygenated deep waters. And since the sinking speed, they basically determine that while fixing nitrogen, what kind of oxygen and nitrate concentration they will get. So we can say that the interacting effects of particle sinking speed and concentrations of oxygen and nitrate in the water column basically will affect the nitrogen fixation rates. But again, this is just a single type of oxygen nitrate concentration in the water column. But around the globe, of course, there will be different types of concentrations. So to check that one, we considered 3 different types of vertical profiles of oxygen nitrate. Like for example, in the first one, this is the oxygen minimum zone, where we see that around 200 to 600 meter, so oxygen concentration is really low. And in the second case, we consider the similar upwelling region where around 200 to 600, so oxygen concentration is around 60 to micromole per liter. And another one is the open ocean, where always throughout the vertical water column, so the oxygen concentration is really high. And we allow different size class of particles to sink there and measure and calculated the nitrogen fixation rates. Interestingly, of course, in all in oxygen minimum zone and upwelling zone, they particles, they fix nitrogen. But interestingly, in the open ocean, where throughout the water column the oxygen concentration is really high, they are also, I mean, the particles, they can fix nitrogen, because I mean, although the outside the particle the oxygen concentration is really high, but because of bacterial activity they can get anoxic interior where they can perform nitrogen fixation, although the nitrogen fixation rate is small compared to the other 2 cases. When you look at the nitrogen fixation rates in the aphotic ocean, so it lies between some kind of 0 to 0.89 micromole per cubic meter per day, so which is almost similar to the rates that we found here. So you can say that our model nitrogen fixation rates are comparable with the bulk nitrogen fixation rates measured in the aphotic ocean, where we don't expect generally the cyanobacterial nitrogen fixers. Only we can expect that the hydrophobic nitrogen fixers will be there in the aphotic ocean. When we look at the depth integrated nitrogen fixation rates, so we see the range of depth integrated nitrogen fixation rates lie between 7 to 65 micromoles per meter per day. And in natural systems, the evidence of when people measure the nitrogen fixation rates in the regions which is basically dominated by heterotrophic bacteria, the range is almost like 6 to 193 micromole per square meter per day. And not only heterotrophic bacteria, when you look at the cyanobacterial nitrogen fixation rates, in most of the regions of the global evolution, it lies between 1 to 100 micromole N per square meter per day. So our rates are almost similar to the cyanobacterial nitrogen fixation as well as the heterotrophic nitrogen fixation. So we can say that the particle associated nitrogen fixation can account for a significant fraction of water column nitrogen fixation. So as a summary, you can say that high bacterial respiration, they create anoxic interior inside the particle where bacteria are able to do nitrogen fixation. Large particles, they have a high chance of getting that anoxic interior, and that's what they can perform, they are suitable for performing nitrogen fixation. Sulfur reduction is more important for nitrogen fixation in sinking particles than nitrate reduction. And nitrogen fixation can occur in well-oxygenated waters. These heterotrophic nitrogen fixation rates, they are significant compared to the other cyanobacterial nitrogen fixers. And overall, as a conclusion, we can say that this particle-associated heterotrophic nitrogen fixation, they contribute significantly to oceanic nitrogen fixation. Yeah, and thank you very much for your attention.
Thank you very much. Excellent. Very intriguing stuff. Let me just have a look at the participants, and we should check the chat first and say if anybody has any questions, please do add to the chat, or just alternatively just raise your hand and I shall spot you in the participant list. Let me just have a quick look. So I've got a quick one while people are contemplating their questions. Looking at the model results where you showed the various properties as a function of time and radius, it wasn't apparent that there was a steady state. I mean, is there a steady state and equilibrium within these particles, or does it tend to be sort of an ephemeral just burning through of the resources that go down with the particle?
Yeah, so it is basically, I start with initial concentration of like polysaccharide and polypeptide, and there is no steady state because they will degrade and they will take material, and of course at some point when it will at some point reach to the steady state, because I assume that a fraction of the polysaccharide and polypeptide, they are labile, and when they will run out of all those labile material, then they will, they will not do anything. Only the bacteria will start dying, and they can't do anything. They can't survive there. But until then, they will keep degrading all those materials, and if the conditions persist are favorable, they will perform nitrogen fixation.
That's interesting. I mean, related to that, I mean, I guess this is an entirely different topic, but do you have a feel for the identity of the bacteria, because it seems quite a suicidal life history in the terms of you put yourself in a particle and head down, and so that would imply that they're at least able to sustain themselves some of the time in open water prior to the formation of particles that leave the surface. So I just wondered if there's potentially an overlap between the nitrogen fixing communities.
Yeah, I mean, it's really a big question, because eventually the particle will sink and reach to the bottom, right? It's kind of suicidal, but I guess in the apodic zone also we find that the presence of this bacteria, and they can perform nitrogen fixation. So I mean, it's not like that they will die properly, but of course, I mean, maybe the community is different there, or maybe only it's not like the particle they will sink, but there are several other processes like fragmentation. I mean, because of that, some bacteria can go out, and they can leave also as well as. But of course, I mean, a number of bacteria will die. That's their life strategy or stages.
Yeah, interesting. And do you have any feel for how you could scale it up? To put into a global model, for example, because I guess people who put nitrogen, euphotic nitrogen fixation.
So at the moment I'm working on that one. I mean, I considered the nitrate and oxygen profiles in the global ocean, and then put the model there. And of course, I did a few modification in the model, because at the moment, like when particles sink, they degrade materials, but the size is fixed at the moment. But now I incorporate that part, because I mean, this is the first model. That's why I didn't want to make it more complicated. Just made it very simple. But at the moment now, I incorporated that thing. I mean, when the degrade material, the size will also sink depending on the available carbon inside the particle. So and several other modifications also in the model, and I have the basic the global results, and
Yeah, fascinating. Yeah.
Mm-hmm.
Yeah, I hadn't occurred to me until that point, but you're right in terms of, you know, having a particle size which changes with sort of time and depth is obviously going to be an important part of capturing the dynamic. Yeah. Oh, very interesting, very interesting. Sorry, I'm hugging all the questions. Let me see if anybody else has anything. Yes, so we have one from, oh, we've got a couple coming in. Mar, do you want to ask your question?
Yeah, so you are right. You are right. I mean, yeah.
You read it already. Yeah. So I was looking at the paper, and I see it goes up to, it's in micrograms per nitrogen and cell per day, which is, yeah, I have to do the math, but usually like per single cell, I think we do femtomole nitrogen per cell an hour or something like that. So how you, whatever, but do you have a feel of how this is compared to previous single cell nitrogen fixation rates?
Yeah, I mean, of course, I compared the nitrogen fixation rates with the available nitrogen fixation rates per cell. I mean, and I should say that they match quite. Actually, the maximum nitrogen fixation rate is taken from like some of the papers. So it can't exceed that rate. So they are quite realistic.
Okay. Thank you.
Yeah, and it's from an experimental result.
Thank you.
Thank you, Mar. Uta, I see you've got a comment. Do you want to make your comment?
Hi, yeah. It wasn't a question. It was more a comment to you asking what the bacteria would benefit to falling all the way to depth. Maybe we need to think about these aggregates as, you know, not going all the way to depths, but forming and reforming and bacteria jumping on and off, or organisms in general. So maybe that's not such a big concern. Not sure, just a comment out there.
No, I agree. I completely agree. I mean, there are also papers like, I mean, they work on this, breaking and aggregation and deformation of those particles while sinking. Yeah, so I mean, in future, of course, I will try to add those components in the model. But since, as I mentioned, that this is like the beginning of this type of work, and people don't know much about it. That's why I tried to make the model as simple as possible, although this is already very complicated, and it takes already much time to run that one. So that's why I tried to keep that one. I didn't incorporate those fragmentation and that part in the model. But in future, definitely I would like to get the actual rates. So I would like to incorporate that. So by adding some term in the model, I would like to do that. Yeah, thank you.
Got the thumbs up from Uta. Any more questions or comments? Let me just check in the chat. Okay, can't see anything. Let me just see if there are any. Oh, I see that Mars just put a number into. Oh yes, so it's comparable. There you go. A nice bit of.
Yeah. So as I mentioned that, I mean, I took the maximum nitrogen fixation rate from one of the measured rates. So it can't exceed that rate. So it can be, can be lower than that rate. So it should be, I mean, in the realistic range.
Excellent. So I don't see, oh, a second, I think that might be Mars' comment down there. Yes, so that's Mars. Thank you, Mar. We have had two excellent talks. Thank you very much to both the speakers.